And good afternoon, everyone that is here. So, such a special day for all of us, right? So I've never done three presentations back to back, so we will hopefully get over this. So there's food and coffee available for the ones that joined us over here. So it's with a lot of pleasure that I would like to introduce you to George Hutchinson for the ones that George is a mechanical engineer, graduated in 21 from UCF and had joined my program as one of the first students that I had. And I had done amazing work as you all are going to see in a second. So I would like to really bring you to enjoy the opportunity to listen what George did over the last 2 years. And then we will move forward with a lot of questions, hopefully for you. So, George, so the floor is yours.
Thank you very much.
Please enlighten us with.
Just share properly.
There you go.
And then presentation up. All right, I think we're good. Everybody can see it.
Can everyone on Zoom see us nicely?
Yes, we can see it.
Okay, thank you.
Thank you.
All right, so let's get started. Welcome everybody. Thank you guys for coming. I'm happy to kick off our slate of excellent seminars today. And I'm going to be talking about some approaches that we did to maximize the sustainability of CEA generally, but leafy green and strawberry production specifically. So we're going to start with some introduction. And first, just briefly going over where strawberries are produced in America currently. The majority is in California, as you can see on the right, throughout the year with some Florida contributions in the winter. And then we can see the major production regions on the left up in the central coast by Salinas, and then moving down the southern coast as well. We have some production. Same idea for lettuce here, going over major production areas. We have the central coast of California, again around Salinas in the summer, and then in the winter, the production moves down around Yuma and the Imperial Valley in Arizona, Yuma. And so why do we care about this? Climate change, obviously, is coming. And the Southwest is prone to droughts and heat waves. And right now, we're going to take a look at those projected changes to yearly heat. So the map that you see in front of you, the different colors are for the number of days out of the year on average that the temperature is above 30 degrees Celsius. And we have some of the production areas highlighted in green boxes here. So right now you're looking at the average for the last 30 or 40 years from 1980 to 2009. And then under the medium emissions scenario by mid-century, these are the changes. So we see some change, especially around Yuma and along the coast. We see moderate changes, but if we don't get our act together and reduce emissions, then that is going to be the future by mid-century. So this is obviously very bad for especially lettuce production around Arizona in the winter that relies on the cool desert winters to produce lettuce outside. So, and it's not only the average heat that's a problem for climate change. Extreme weather is projected to increase in intensity, frequency, and length. So meaning that droughts are going to be more intense and more frequent, and so are storms and heat waves. This is really bad for outdoor agriculture. So controlled environment agriculture produces a solution to this and can contribute to the security of our food system by protecting the crops from extreme weather and also the associated labor, because people need to harvest and plant the crops as well, and they can't do that if the fields are flooded or if there's a really bad heat wave. CEA also provides a couple other advantages, such as water and space efficient production, and it limits fertilizer use as well. But there still remains a question, is controlled environment agriculture sustainable from a climate change perspective? So does it release fewer greenhouse gases than field emissions? And it's kind of a mixed bag answer from all of these sources at the bottom. So most of them agree that the source of energy is the main contributing factor to whether it's sustainable. But obviously we can't address that energy production from a horticultural perspective. So we're going to take the avenue of addressing the inherent sustainability of controlled environment agriculture to try and increase that and mitigate the negative effect of CEA on the environment. So that's the project objective. We're going to try and maximize the sustainability of CEA and get more outputs from fewer inputs. The objectives are to execute system comparison experiments for the first two chapters. Chapter 1 is strawberries, chapter 2 is arugula and lettuce, and then chapter 3, we're going to look at different fertigation strategies for strawberries. Very briefly, we're going to go over the hydroponic systems that we're going to be talking about. In aeroponics, you use a high pressure pump to mist the nutrient solution onto the exposed roots. Moving to the right, in deep water culture, the solution is stagnant and the plants are grown on a floating raft on that stagnant solution, and the solution is aerated as well. In substrate drip, on the bottom left, there is an inert substrate into which the plant's roots grow, and nutrient solution is delivered via drip irrigation. Nutrient film technique is to the right of that, and plants are grown in sloped channels. Solution is pumped to the upslope side of the channel, and then the solution runs down the channel over the plant roots, and the plants can uptake water and nutrients. And then in the vertical tower system on the right, there is a reservoir in green on the bottom. The solution is pumped up a central column, hits the top, and then can percolate all the way down around the tower, hitting all of the plant roots on its way down. All right, now that we've got the introduction out of the way, I'm going to move on to chapter 1. And the title for chapter 1 is a bit of a spoiler alert. The substrate system definitely did better than the 3 water culture systems for the strawberry production. And we're going to find out why. First, we're going to go over the knowledge gap that we identified in the literature, was that substrate systems and water culture systems, there was a lack of comparison between the two for greenhouse strawberry production in hydroponics, and especially comparisons that were at scale and considered resource use efficiencies. So with this gap in mind, we formulated the objectives for the chapter, which was to grow strawberries in substrate and water culture systems, track yield and resource use, and then finally make a system recommendation based on these parameters. Brief overview of the experiment. We had 4 different hydroponic systems. We used 2 different cultivars, Florida Brilliance and Florida Beauty, from December through April, and then we used the Yamazaki nutrient solution. Some pictures of the systems here. So this is the substrate system. It was a 1-to-1 mix of coarse perlite and a bark-based mix, and drip fertigation, like I mentioned earlier. And the way it was fertigated was 1 fertigation event per day with a 20 to 30% leachate target. So this is kind of the standard practice in production settings for this system. This is a picture of the NFT system. We constructed it in-house using vinyl fence posts. Yeah, that's pretty much it for that. And then this is the vertical system. You can see on the left before and on the right after we installed lights. So we did use supplemental lighting for this experiment. Since it took place during winter, we needed to raise the DLI on average to provide plants with adequate lighting. And then finally, on the left, you see the aeroponics system. And then on the right, you can kind of get a macro view of all of the systems within the greenhouse, all lit up with the lighting installed. A couple more pictures here just to orient. On the left is the substrate system, and on the right is the 3 water culture systems. These were the measurements that we did. Balanced EC and pH in the reservoirs, tracked volume, and then we did weekly fruit harvests as well, where we quantified biomass and a couple of other things, and then did a final plant harvest as well. So these are the yield results. And we can see straight off the bat that the substrate system definitely produced higher yields in both cultivars, but much higher yields in the Florida Brilliance cultivar. And before we get into maybe why that was the case, we're going to look a little bit more closely at the fruit quality results. So on the top left, you have the dry fruit biomass, and then the marketable yield. And on the right, you have fruit water content, and then total soluble solids. So we see that the yield, the fruit quality results for the substrate system for dry fruit biomass and marketable yield follow very closely to the total yield results. And then on the right, we see that the substrate system had reduced total soluble solids. And total soluble solids is a proxy to measure how much sugar is in the fruit, basically. So it appears initially that these fruit are less sweet and maybe you would think lower quality, but actually it's because these fruit were more well-developed and had a higher water content. So that sugar that was in the fruit was more diluted. However, the fruits were more well-developed. Now, looking at the biomass results, it becomes pretty clear why the substrate system resulted in higher fruit yield. because for height on the top left, leaf area, fresh shoot biomass on the top right, and dry shoot biomass on the bottom right, the substrate system blew every other system out of the water pretty much. So because it had this higher vegetative biomass, higher leaf area, it could intercept more light, photosynthesize more, and it had more energy to produce fruit. Now, one explanation for why this biomass was higher was that the roots provided more mechanical strength in the substrate system compared to the water culture systems. So the roots could spread out and provide more stability to the crown and the canopy to grow more vegetatively. Rather than the water culture system, they can't push against a liquid and provide support to the canopy. So that's one explanation for why these biomass parameters were higher in the substrate system. Looking at the macronutrient foliar analyses, there's not really any huge significant differences or trends between the cultivars that emerge. But in the micronutrient results, we see one thing that kind of jumps out, and that's the iron concentrations in the leaves from the substrate system were significantly higher than in the 3 water culture systems. So iron is really important from a biological, physiological perspective because it's involved in several steps along the electron transport chain. It's involved in photosystem 2, cytochrome B6F, and of course ferredoxin, which is the ultimate receiver of that electron. And so it's possible that this higher iron concentration lubricated the photosynthetic machinery and allowed the plants to function better and produce more fruit. Finally, looking at the resource use efficiencies, on the top left, we have the harvest index, and then below that is the area use efficiency, so that's the yield per square meter. On the top right, we have the water use efficiency, and on the bottom right, we have energy use efficiency. So we can see that the substrate system on the right had high water use efficiency and extremely high energy use efficiency. And energy use efficiency makes sense because the substrate can hold on to the nutrient solution for the plant to uptake over time, so you don't need a pump running so frequently to provide that nutrient solution. Another thing that kind of jumps out is for the area use efficiency, the vertical system actually did very well comparatively. So even though it had lower yield on a per plant basis, because the vertical system can fit so many plants in one area, it still provided excellent area use efficiency, so yield per square meter. So that to me shows that the vertical system has potential for commercial applications, provided some tweaking essentially, but in most cases, the substrate system is going to be your most optimal system for growth, and the NFT system is really not suitable in the form that we chose. So that's it for chapter 1. We'll move on to chapter 2 now. And this was the lettuce and arugula system comparison. So the knowledge gap that we identified was that there had been some system comparisons before, but they were not very large in scope. They only took into account maybe 2 water culture systems and 1 resource use efficiency. And so this gap was testing 3 or more systems and considering multiple resource use efficiencies. So that was the goal. And again, it's very similar to chapter 1, multiple systems, multiple resource use efficiencies, and making a system recommendation for each of these crops. Quick overview. We used KC lettuce and Astro arugula in the 4 water culture systems were deep water, nutrient film, vertical, and aeroponic. And then an important thing to note is that it took place during the summer from June 15th to July 13th. So it was hot in that greenhouse during this time. So we used 2 heat-resistant cultivars. And it was a 28-day experiment, but arugula was harvested at 21 days. So these are some pictures of the system. This is the deep water culture system at transplant. And then 19 days later. And we have the NFT system at transplant. And 19 days later as well. Vertical system, same thing. 19 days later. And then finally, we have the aeroponic system at transplant and 19 days later. These were the measurements that we took. So we had non-destructive measurements, height, chlorophyll, anthocyanin, destructive plant measurements, all the standard stuff, and then system measurements as well. Very similar to the last one with the addition of solution temperature. So I had heard from one of our industry supporters that his system, the vertical system, had reduced solution temperature. It tended to reduce solution temperature. So I decided to install thermocouples and use a data logger to automatically monitor solution temperature throughout the experiment. And he was right. You can see that the vertical system had reduced solution temperature, an average solution temperature that was lower than all of the other systems throughout the experiment. And this makes sense if you think about how it works. The solution is dripping down the column, it's interacting with the air, it's evaporating, so it's cooling as it falls down, and then over time that effect builds up and cools the entire reservoir. Something else that was interesting in these results is that the deep water culture system resulted in the highest average temperature for both crops as well. This also makes sense because the solution is stagnant, it's flat, it's spread out, has a high surface area to volume ratio, so it heats very easily, and it's also in a big black tub exposed to the sun, so that heats up as well, heating the solution. These temperatures are going to be important later on. Looking at the non-destructive measurements, we have arugula height on the left, and then chlorophyll and anthocyanin content on the right. And we start to see the trend emerge. That's going to be kind of one of the themes of this experiment, was that arugula in the deep water culture system behaved differently than the other three systems. So we see on the left that the arugula plants in the deep water were shorter, and they had higher chlorophyll and anthocyanin content compared to the other three recirculating systems. And then we can also see on the right that there wasn't a whole lot of difference for lettuce in the chlorophyll and anthocyanin contents. Looking at the yield, this trend continues. But first, we see that the three crops behaved very similarly for the three water culture, or for the three recirculating systems. We have the nutrient film technique with the highest, then aeroponic, then vertical in descending order. However, we see here that the crops had very different responses to the deep water culture system, with arugula having the lowest yield in the deep water culture and lettuce having the highest yield. Looking at some more biomass results, we have fresh and dry root biomasses on the left, and then on the right, we have leaf area and dry shoot biomass. Some similar trends to the overall yield. Nothing too crazy jumps out, except for the dry root biomass was really high compared to the other three systems in the aeroponic system for arugula. And this matches some previous research with other crops that have indicated that root development and root growth is spurred in aeroponics compared to other systems. Moving on to the macronutrient results, we begin to see an explanation for why we observe these differences in yield from the deep water culture system. If you notice that both crops had reduced macronutrient concentrations in the deep water culture system compared to the other three recirculating systems. And why this happened, it could be because the nutrient solution isn't recirculating in the deep water culture system, there could be transient depletions of nutrients around the root zone that can only be rectified via diffusion, which is a relatively slow process compared to just moving the nutrient solution over the roots. So over time, as the crop matures, there's more of this depletion, the depletion zones grow larger, and then the plants have less access to nutrients overall. And then something else that was interesting was that the arugula from the aeroponic system had a higher concentration of sulfur in the leaves. And so arugula produces a secondary metabolite called, a class of secondary metabolites called glucosinolates. And these are part of the compounds that give arugula this bitter flavor that we all love, hopefully. And so arugula from the aeroponic system could have had higher concentrations of glucosinolates. So this definitely merits further investigation. Looking at the resource use efficiencies, and we have shoot water content as well. And shoot water content is important because we see that arugula had reduced shoot water content in the deep water culture system. So this, to me, indicates that those plants were suffering from stress and less able to osmoregulate and uptake and sequester water in their cells. And so that's why, they had lower nutrient concentrations. That's probably a byproduct of the heat stress from the higher solution temperature. And we see support for that with the shoot water content being reduced. Below that, we have the water use efficiency. We see that there are similar trends to the overall yield in water use efficiency, but there's some influence from the water use from each system as well. And then for the two on the right, the energy and area use efficiencies, we see extremely similar trends for both between the two crops for both parameters. And these indicate that both of these parameters are more system driven rather than crop performance driven, yield driven. And so that's part of the conclusion for this. I made this nice table to synthesize some of these results. So we can see in the top table that the aeroponic system for arugula produced excellent yield, water use efficiency, and energy use efficiency. Whereas for lettuce, the deep water culture system produced excellent yield and water use efficiency. Now, a caveat to these results, like I mentioned before, is that energy and area use efficiency are very system driven and liable to change as systems scale. Whereas yield and water use efficiency are more inherent system qualities. And that's it for chapter 2. Last one, bear with me here. So we're going to look at some different fertigation strategies for strawberries in hydroponics. So these are 3 of the major strategies that currently are popular for hydroponics in general, but also strawberries specifically. You can use a timer to automate your fertigation. I mean, that's very simple, but it's not super targeted. One of the common production practices is to use a leaching fraction approach where you apply the fertigation solution regularly, but at a volume that results in 20 to 30% of that volume leaching through the solution. And then finally, sensor automation is something that's beginning to be explored and has been explored in other crops to good ends, such as resource saving, crop quality improvements, and biomass improvements as well. So the knowledge gap that we identified was that all 3 of these strategies had not been compared before for strawberries in hydroponics. So that was basically the goal of this experiment, was to compare all 3 of these strategies. And again, similar theme, track yields and resource use, and make a recommendation for fertigation strategy for these for this crop for strawberries. A little bit more info. This is kind of a rehash from before. For the substrate system, troughs, drip irrigation, 1-to-1 of these 2 products right here. Here's a picture of the system, same supplemental lighting, same setup. And for the treatments, like I said, there are 3 strategies. So there's the leaching fraction fertigation, which is manually controlled at regular intervals to get 20 to 30% leachate through the solution or through the substrate. There's timer fertigation, which I played with until I arrived at 3 minutes every 3 hours, which was a reasonable amount of application. And then we had 4 automatic fertigation treatments at 4 different volumetric water content thresholds. And these thresholds were determined from the substrate container capacity at 0.42. And the thresholds were 0.36, 0.30, 0.225, and 0.15. So 0.36 being the wettest and 0.15 being the driest. And these were constant thresholds. So a little bit more about the control system for this, for these automatic fertigation treatments. We had these soil moisture sensors, which sent a volumetric water content reading to the data logger. And the data logger took this information, and if the average of the 4 replications was below the threshold, then the data logger sends an activation signal to the relay. And then the relay would, relay the power on signal to the pumps, and the pumps would deliver fertigation solution to the plants. And this happened every 3 minutes throughout the entire 129-day experiment. So once again, we have these 6 fertigation strategies. We used the same cultivars, and it was December through April with the modified Yamazaki nutrient solution as well. So these are the results of the sensor automation. This is a graph of the volumetric water content data from all 6 treatments throughout the experiment. So, and the dashed lines represent the different thresholds as well. So we can see that the automation treatments, the automated sensor-based treatments, adhered very closely to the threshold lines, indicating that our automation and our goal of applying a constant threshold was successful. And then you can see the volumetric water content results from the leach and the timer fractions as well. The leach is in gray and the timer is in pink. So that's just kind of supplemental data tracked for consistency. We didn't use that data to make any decisions or anything. So looking at the yield, slightly different responses between the two cultivars for all of the treatments. However, one thing kind of jumps out is that this 0.225 treatment resulted in the highest yield in both cultivars. And before we get into why that may have been the case, we're going to look a little bit closer at the fruit quality results. So we have marketable yield and fruit drying biomass on the left, and then total soluble sugars and fruit water content on the right. And again, on the left with those two graphs, we see very similar kind of trends to the overall yield. And then on the right, we see that the timer treatment resulted in high total soluble sugars in the fruit. But again, this is due to reduced fruit water content. So kind of a similar idea as the first chapter, where if the fruit water content is different, the total soluble sugars are going to be different as well. And that doesn't necessarily mean that the fruit is of better or worse quality. With the biomass results, the point I want to make here is that just because this treatment, the 0.225 treatment, resulted in the highest yield, it doesn't mean that it had the highest fresh shoot biomass or the highest dry shoot biomass. And it also didn't have the tallest plants or the plants with the most leaf area. So why the heck did this treatment produce the highest yields out of all of the treatments if it didn't have the highest biomass? Because you would think if you have higher leaf area, once again, you can make more photosynthetes, you can intercept more light, and maybe produce more fruit. But we start to see a trend emerge in the micronutrient results. That treatment resulted in very high manganese and iron foliar concentrations for the Florida Brilliance cultivar, and same exact thing for the Florida Beauty cultivar as well. And I elaborated on iron before, how it's important to the photosynthetic processes. Manganese is vital as well for a couple different things. One being that it's a cofactor for a lot of enzymes in the citric acid cycle. So once again, these higher concentrations could have kind of lubricated the biochemical machinery to produce higher fruit yields. Looking at the resource use efficiencies as well. On the left, we have water use efficiency, and on the top right, we have harvest index, and on the bottom right, we have energy use efficiency. And so we see various kind of similar trends for yield, and we see good performance from the 0.225 treatment for both water use efficiency and harvest index, which is kind of biomass use efficiency almost. And then the only caveat being it had lower energy use efficiency, especially for the Florida Brilliance cultivar. So the takeaways from this were that the sensor automated fertigation worked and produced good results for the two drier of the treatments. And it's possible that optimal volumetric water content in the substrate could enhance micronutrient uptake and accumulation. So we're done with 3 chapters. Let's move on to the conclusions. And it's short and sweet right here. Strawberries are most optimally grown in substrate. Lettuce is deep water culture, and arugula in aeroponics. And then sensor automation does show potential for hydroponic strawberry production, but it definitely merits further investigation, maybe messing with more granular thresholds or multiple thresholds instead of a single threshold. So those are kind of directions that I see investigations going in. I'd like to thank all of our industry supporters for donating equipment or providing guidance in how I executed these experiments. Paul and Marilyn Bretlinger from Crop King, Brett Boland from Tower Farms, Pete Stefanos from Haygrove, Roger Velo from Aero Farms, Frederick Ford from Lareau, and then nice folks at Florida Strawberry Growers Association as well. They all were very kind and very willing to help, so I appreciate you all for doing that. And then finally, I couldn't have got here without the support of a ton of wonderful people. Most of the people in this room, everybody in the lab, everybody in Miller, Lynn, thanks a lot. My whole family, all my friends, I couldn't have got nearly this far without all of you. So thank you all very much. And with that, I'm done. And I'd love to take some questions.
Sure, there are some posts on the chat, so there's since the beginning, so I don't know if those are questions or not. But if you could just look in the chat, it'll be great. Nope. Okay, fine.
Yeah, if you have any questions on Zoom, feel free to shout out.
Hey, George, this is Rebecca. I have a question for you.
Yeah, go ahead, Rebecca.
So for the strawberries, you found that in substrate, the soluble solid content was lower, and you said that was because the fruits were larger, had a higher water content. But do you know if that soluble solid content is still marketable for those substrate grown strawberries?
Yes, it is for sure. Some field trials for both of those cultivars showed total soluble sugars between, I think it was like 5.7 and 8.2, and ours from the substrate system were right in that range. So those are definitely marketable as well.
For your second chapter, George, you did find different crop responses to different systems. So translating this into something more practical, so would you recommend in a grower, a commercial grower, having multiple systems in their facilities for producing multiple crops?
Yeah, I would say so. You know, if the crops respond significantly differently like that and have significantly different resource uses, then I would say, yeah, there's definitely going to be a learning curve. It's going to be challenging to implement multiple systems, but I think over a longer period of time, that will be worth it in the end because the crops will respond better and have higher quality under different systems. Yeah.
That's kind of a question, but in your first chapter, you found that in the substrate systems, you had, I think it was like 5 times as much iron in that particular system. Did you find anything or have any thoughts on like what mechanism or why that would be possible in that particular system?
I mean, so in substrate systems, you have cation exchange capacity that you don't necessarily have to as great of a degree in water culture systems. So the substrate could have held on to that iron to provide a longer, larger proportional uptake over time. That's kind of the one thought that I had. The other thing is that the plants in that system were more able to properly osmoregulate, and thus they were able to not only uptake but sequester that iron as well.
You mentioned you did some photosynthesis gas exchange measurements. Did you not find anything to support the conclusions?
No, there was nothing super useful in the gas exchange measurements, unfortunately. There was only one significant difference, but that could be attributed to user error for sure. So yeah, that's why I didn't present those results.
Can I ask a quick question from Griffin? Yeah, really great presentation. I enjoyed it. Regarding the strawberry irrigation, would you say that if a grower was using just a timer, did you find enough to really recommend that they would switch to a sensor-based system, or would you say probably just stick with your timer system for now?
I mean, there's kind of two sides to that coin. One is, if it ain't broke, don't fix it, right? If it's working and you're satisfied with your performance and you're not wasting a lot of water or time or energy implementing that system or implementing that fertigation strategy, then why change it, right? I would recommend maybe on a small scale experimenting with some sensor-based strategies to see if you can really dial in your fertilizer application and maybe increase your fruit quality or even increase yield or something. But definitely not, switching the whole facility from one to another overnight. That's never a good idea, I don't think. So I think some experimentation, and every system is different as well, and every substrate is different and will respond differently. So definitely testing on a small scale before wider implementation would be recommended. Yeah.
Great, thank you.
Yeah. Anything else from Zoom?
All right. Thank you very much.
All right.
I appreciate it.